Hello, welcome to Vedial Vagaparai. In this video, we are going to see about ribose sugar. This particular topic is based on a JEE Main 2025 question, which appeared in the morning shift of 24th January. So, ribose sugar is a sugar of the nucleic acids, DNA and RNA. So, we will see in detail the basic information about ribose sugar, and then we will look at the question also. So let's see the question first. So the question has four options and there are five properties of the sugar that is listed here. The question is the carbohydrate ribose present in DNA is a pentose sugar present in pyranose form, in D configuration, a reducing sugar when free, in alpha anomeric form. So they have given five different properties of this ribose sugar and they've asked us to choose which among the following is correct. So we will first see the ribose sugar and its structure and all these parameters concerning the ribose sugar. The type of ribose sugar that is present in RNA and DNA, and then we will come back to this particular question. So let's first take the structure. So when we see the structure of the ribose sugar, we know it is a 5 carbon containing sugar and it is an aldehyde. So this is a pentose sugar because it is having 5 carbon in the carbon chain. And then all sugars, they are given a configuration. So the configuration of carbohydrates, all the carbohydrates are designated based on the OH group from bottom. So the first OH group from bottom, if it is written on the right hand side, then the sugar is called as a D sugar. So all naturally occurring sugars, the monosaccharides and disaccharides that we learn are all D sugars in the sense from bottom, the first OH is written on the right hand side. So likewise, ribose sugar is also a D sugar because the OH is on the right hand side. And it's a very easy structure which is easy for us to remember. All the OH are on the same side of this particular molecule. So next what we will see is about the cyclic structure of these carbohydrates. So we must know that these carbohydrates have a very beautiful property of forming an intramolecular ring system or a hemiacetal formation, in the sense because the carbohydrates contain a hydroxyl group and a carbonyl group. The hydroxyl group and a carbonyl group can or in other words, it is an alcohol and an aldehyde or a ketonic group. Here it is an aldehyde group. So the alcohol and aldehyde can combine and form hemiacetal. So all carbohydrates, be it hexose or pentose, they can form either 6-membered ring or 5-membered ring by forming hemiacetal. So this molecule, this structure is a hemiacetal structure, and the formation of a hemiacetal structure leads to two ring systems. One is called as a furanose ring, wherein it is a 5-membered ring structure. Another one is the pyranose ring, which is a 6-membered ring structure. We will know that glucose is a carbohydrate which forms a 6-membered pyranose ring structure. Fructose is a ketone, so it will form a furanose ring structure or a 5-membered ring structure. Ribose by itself is only a 5-carbon containing compound. So the possibility of this fifth OH to react with the aldehyde will give you a pyranose structure. The fourth OH can react with the oxygen and form a furanose structure. So we know that 5-membered ring or 6-membered ring are stable structures, and that is the reason why these two possibilities are there for carbohydrates when they are in solution. So this ability of a carbohydrate to change between the two open chain and closed chain structures, this is called as mutarotation. And if you are interested in learning about glucose and other disaccharides, you can look at our earlier videos on carbohydrates, part 1 and part 2, where we have discussed in detail all these open chain and cyclic structures and various properties of carbohydrates. So now coming back to ribose. So the next important property is it is a reducing sugar when free. Again, what is a reducing sugar? Again, this definition basics is already there in our earlier videos. Please go and see those videos for a detailed explanation of what is a reducing sugar. Here I will just give you a simple reason, that is these carbohydrates have an aldehyde group. So these aldehyde group can be oxidized to carboxylic acid when reagents like Tollens reagent are used. So when a carbohydrate is treated with Tollens reagent, silver is precipitated. Okay, so in a sense, silver in silver nitrate is precipitated as silver element, and that is why it is reduced. So these carbohydrates are called as reducing sugar, which reduces the silver in silver Tollens reagent. So on the other hand, these carbohydrates will become oxidized. So this possibility is there only when the aldehyde group is free and it can react. So whenever these carbohydrates are in a cyclic system, the aldehyde group is not free. The aldehyde group is converted into a hemiacetal. So when during reaction, this cyclic structure will open up to the open chain and then it will react with Tollens reagent. But suppose if this molecule is interacted with another molecule and it is locked in glycosidic linkage, then it cannot. Only in those times it is called as a non-reducing sugar. Please go and see our earlier videos to benefit from them to understand reducing and non-reducing sugars. Detailed explanations are given there. Here, this is not the context of discussion, so I am stopping it here. So here what we want to see is ribose sugar has the possibility of forming a pyranose ring and a furanose ring, and which is the ring system that is more stable in case of ribose. So pyranose ring is with this primary alcohol, rather ribose forms a furanose ring only. And again, because it can open up for reaction in this, when it is in a free state, then ribose is a reducing sugar. So when free is a very important thing. When this portion is reacted upon, then this ring system will not open. Okay, so there it is a non-reducing sugar, whereas when it is present as D-ribose without any reactions, a reaction changing its structure, then it is a reducing sugar. So next what we will see is about the furanose form. So as I told you, between the two possibility, ribose, because it's a 5 carbon compound, this will predominantly form a furanose ring system only and not a pyranose. Another point to note is this OH group. So here the OH is not given as a solid arrow, sorry, solid bond. Rather, it is given as a oblique bond. In the sense, this OH has two different orientations that are possible. So this is a general structure. So now what we are going to see is what would happen to this particular OH group in when the ribose sugar is cyclized. So let us see the two possibilities. So one possibility is the OH attaches here and the carbonyl oxygen is formed on the left hand side. Another possibility is the OH this group is oriented in this side, and so when OH attacks, this OH is on the right hand side. So there are two possibilities of cyclization. One where the OH is on the right hand side, another where the OH is on the left hand side. But then when we are representing these structures, we know that in case of carbohydrates, this representation is called as the Harvard structure. So in the Harvard structure, we see when the OH is after cyclization, when the OH is written down, then it is a alpha. This OH that is there at the first carbon, this is a newly formed stereocenter, and this is a anomeric carbon. And this is a newly formed anomeric carbon, and this anomeric carbon is making the molecule chiral, and that is the reason why you are getting two possibilities. One possibility where the OH is down, another possibility where the OH is up. The possibility where the OH is up is the beta form. The possibility where the OH is down is the alpha form. So there are two anomers are possible when hemiacetals are formed, and these anomers are alpha D ribose and beta D ribose. So in alpha D ribose, all the OH are down, and in beta D, the OH on the anomeric carbon is up. When you compare it with that of the fifth carbon, In alpha, they are opposite each other. In case of beta, they are on the same side. Now let us see the nucleosides. So we know a nucleoside is when a sugar and a base combine together, then it is called as a nucleoside. And then we also saw the alpha and beta anomers of ribose. So the OH would have been here in case of the alpha, the OH would have been here in case of beta. So this is a simple representation for us to understand how the substitution of the bases to the ribose will result in a structure with alpha and beta anomers. And we know popularly we see all the polynucleotides being written in this format. It means the ribose sugar in all the nucleic acids are in the beta anomeric stage, and that is one of the less sterically hindered stage. And then coming to the name of the ribose. So here you see this particular position where the OH is not there. So because the oxygen is absent here at this particular position, this ribose is called as deoxyribose. So now coming to the nucleoside of DNA versus RNA. So the earlier representation for us to understand alpha and beta anomers, this is for us to understand the ribose sugar. So the ribose sugar has only hydrogen here, whereas here there is OH group. So this is ribose and this is not having oxygen. So this is called as deoxyribose, which is present in DNA. And then again, the main difference between RNA and DNA is in case of DNA, we have thymine as the base. In case of RNA, thymine is substituted by uracil. So this simple nucleoside is a nucleoside of the RNA. This is a nucleoside of the DNA. And so here we see the two major differences between DNA RNA. In DNA, the sugar is deoxyribose. In RNA, the sugar is ribose sugar. In DNA, thymine is present. In RNA, thymine is replaced by uracil. So this is the two major difference, and so that is the reason why I wanted to show these two structures. On the other hand, the OH is on top. So in both DNA and RNA, the anomers of the ribose or deoxyribose sugars are beta anomers only. So this is a simple nucleotide. So what is a nucleotide? So base, sugar and phosphate, when it is repeated n number of times, then it becomes a polynucleotide. And here because there is no oxygen, so this is the nucleotide containing deoxyribose. So this is a segment of DNA, and here they have marked out the bases. So here the base is thymine, here the base is adenine, it is cytosine here, and here it is guanine. So all these four bases are the ones which define the genetic code, and we understand protein biosynthesis based on the sequence of these bases in the DNA. So the DNA strand also we see here, it all begins from the 5' end to the 3' end. So if we know it is a double helical structure, so this strand is 5' to 3' end, means the complementary strand will be from the 3' to 5' end. But the scope of DNA structure is not here in this particular topic. I want to show only about the ribose. So the ribose is deoxyribose, and then it is a pentose sugar, and then it is having the beta anomeric structure, and we understand that in a free state it is a reducing sugar, but here in this case this ring cannot open, and so it is not a reducing sugar when it is in the DNA structure. So let's go back to the question. So when we see the question is quite easier to solve now. So the carbohydrate ribose present in DNA is a pentose sugar. We all know it's a pentose sugar, and it is present in the pyranose form is a wrong option because it is present only in the furanous form. It's a 5-membered ring. And in D configuration, yes, all carbohydrates are D configuration. Reducing sugar when free, yes, when this group is not attached, instead if there is a OH group, then it can open, and so it is reducing sugar when free. And in case, in the last case, it is an alpha anomeric form. No, here it is in the beta anomeric form. If it was alpha, the base must be at the bottom. So this is the option that we have seen. And so the correct option which gives you the solution is A, C and D, B and E are wrong answers. Thank you. Like and subscribe to our videos for more videos. Please visit this channel and patronize it. Kindly share with anybody who is in need of these videos. Thank you.